next we'll be starting with the next module, which is module number 8, which is related to pharmacovigilance. So now we'll start with the next module, that is pharmacovigilance. Okay. So what do you know about pharmacovigilance? Anyone knows what is pharmacovigilance? So, like pharmacovigilance activities or this process comes after when the drug is approved for the marketing. Okay, so when the drugs enters in the market, then the pharmacovigilance studies, or they are also known as phase 4 clinical trial studies, or post-marketing studies or post-marketing surveillance, but the right term which is used, that is the pharmacovigilance. So now WHO defines pharmacovigilance as a science and activities relating to the detection, assessment, understanding, and prevention of adverse effects of any other medicine or vaccine related problem. Okay, so now the definition of pharmacovigilance as per WHO is that it is pharmacovigilance is the science, or it is a field, or we can say it is a set of activities which are done to detect, assess, and understand and to prevent the adverse effect which are associated with any medicine or vaccine. Okay, so we detect so in this pharmacovigilance, what is done, the professionals, they will detect the adverse event, they will assess the adverse event, and like whether the adverse event is related to the drug or not, and then to understand that adverse event and then prevent that this adverse event should not occur in future. Okay, so these adverse events, whether they are related to medicine or any vaccine or any biological product, so in all the cases pharmacovigilance is required. So it is abbreviated as PHV or PV. So pharmacovigilance aims at improving the quality of patient care. So now what happens, like we all know that these medicines, they are the adverse, these medicines, drugs, they all are having one or more adverse drug reactions. Some drugs, they are having very serious adverse effects. Some are having non-serious adverse effects. With some, the adverse effects are very frequent. With some, the adverse reactions are not so frequent. Or with some drugs, the adverse drug reactions occurs in long term. Okay, like when you continue the drug for a longer period of time, and then you will see the adverse reaction. Some drugs, as you take that, or as that drug is administered at that same, then again then the adverse reactions are seen. That example is with some antibiotic, that when as you take that antibiotic and then someone is allergic to that antibiotic, rashes or redness appears on the skin. So that is the so this is okay. So now we want to so it is not like this, that the patients will not take the drugs. The drug is to be administered to manage or to treat the disease. Okay, so for that purpose, it is always recommended to monitor the adverse effects, and the adverse effects which are already known or which is already predicted. And if it is known that this drug will have these adverse effects, may have, so that should be informed to the patient. Or there are certain activities which are planned so that these adverse reactions can be prevented. Overdosing of the drug should not occur. Any medication error should not occur. So in that case, so that is the case. So by this pharmacovigilance, and what about the adverse drug reactions which are not known? Okay, which we do not know, which we do not know about, which we are not so aware of, or which are not detected at the time of clinical trial. They may be the long-term adverse effects. So for that purpose, it is required that it is always required that the adverse drug reactions, they should be detected during the pharmacovigilance activities. So hence the quality, so that these adverse reactions can be known. And in case if it is known that the adverse reaction with any drug is very frequent and very serious, they can be removed from the market, they can be withdrawn from the market, so that unnecessary harm to the patient should not be caused. So improve the quality of patient care. Increasing patient safety whenever it pertains to the use of medications. So the patient safety can be increased related to the use of medications. Supporting public health programs by providing accurate, unbiased data for determining the risk benefit profile of drugs. So then support the public health programs by there are various public health programs which are conducted by various organization, marketing authorization holder and various companies, so that with this pharmacovigilance activities the accurate unbiased data is presented, so that the risk benefit profile of the drugs can be determined, and the same information can be shared in the aggregate reports as well, so that the regulatory authority can also come to know the risk and the benefit which is associated with the use of the drug, and on the basis of the information which is shared with them related to risk and benefit balance, they can take the decision that whether they should continue the drug in the market or they should take the strict decision to withdraw the drug or completely ban the drug. Okay, so that's why pharmacovigilance is as important as it is in the case as the clinical trials. Industrialized countries have established various safety databases, like EU have UDRA Vigilance, FDA has FDA Adverse Event Reporting System, FERS, etc., to facilitate this work. So now there are all the countries, they have their own database or own way of reporting forms. They give the online forms, offline forms, in which by which the consumer or the healthcare professional, they can report about the adverse drug reactions. So like European Union, they have the platform UDRA Vigilance, and they also have a yellow card scheme. And with that yellow card scheme, the adverse drug reactions are reported. Then FDA Adverse Event Reporting System, FDA have the Adverse Event Reporting System, that is FERS. Our country, India, is also having the reporting system. Like we also provide the reporting form, adverse drug reaction form, and this is managed by Indian Pharmacopoeia Commission. And by reporting these adverse drug reaction, the pharmacovigilance work can be facilitated. As a consequence of a thalidomide incident of 1961, WHO has developed a PV program. So there was a thalidomide incident which occurred around 1961. And in that thalidomide disaster, or it is also named as thalidomide incident, in that there are many pregnant women who has taken the drug just to overcome the morning sickness. But due to the effect of the drug, the baby which was born, they were deformed limbs, crippled limbs, deformed limbs. So that is considered as a thalidomide disaster. And after that thalidomide disaster only, the PV came into existence. Then what are the methods in pharmacovigilance? So what are the methodologies which were used to collect the data in pharmacovigilance related to the adverse drug reaction? So there are 4 important methods in pharmacovigilance, such as passive surveillance, active surveillance, cohort event monitoring, and targeted clinical investigation. So now we will discuss them one by one. First is passive surveillance. The use of spontaneous adverse event reports voluntarily reported to the holder of the marketing authorization or the regulatory body is a form of passive surveillance technique, like spontaneous reporting, prescription event reporting. So that all comes under passive surveillance, in which the consumer or the healthcare professional, they directly report about the adverse drug reaction to the marketing authorization holder, or they themselves fill up the form, the adverse drug reaction form, and submit it, so that the regulatory authority or the concerned authority, they will come to know about these adverse reactions, and a proper analysis can be done on that. Okay, so that is known as a passive surveillance technique. Here, statistics pertaining to the negative reactions are gathered and stored in a national or local database. So now here the reactions or the reports which came, or whatever the reactions are related to the adverse drug reaction, so that all information is stored in national or local database. So every date, like there are certain databases which are available, like UDRA Vigilance, Vigibase, Vigiflow. So these are the name of certain databases which are available. So in that, the list of all the adverse reactions which are related to a particular drug, or the signals which are reported, so that information is stored. The reporter's name is kept secret, but information about the patient, such as the place of origin, gender, age, and comorbidities that they already had, can be retrieved through the reporting forms. So the reporter name, that who reported this particular incident of adverse drug reaction, that is kept secret, but the information about the patient, like who is the patient, where from where to which state, to which place they belong, what is their gender, what is the age, if the patient is suffering from any kind of comorbidities, or what is the condition the patient was suffering from, since when he is taking the drug. Okay, so when and when the start of the onset of adverse drug reaction, whether the adverse drug reaction was reversed or not. So all that information is given in the adverse drug reaction form, and that in adverse and from that reporting form, that information can be retrieved. Okay, the next is active surveillance. So active surveillance, this approach intends to track particular adverse drug events and determine the total number of adverse drug reactions using a pre-planned procedure. It is often referred to as safety monitoring or toxicity monitoring. So there are various safety monitoring studies are done, like going through the registries, and then prescription, going through the registries, and then conducting various surveys and all, so that from there, from those surveys, from interviewing the participants, patients, from safety monitoring, the information can be collected related to that. The information can be collected related to the adverse drug reaction. So now this adverse reporting, these active surveillance, they are the pre-planned procedures, like a full procedure is made according to the plan, that how the plan was, or how this procedure will be conducted and how it will be concluded, and hence it is also referred as a safety monitoring or toxicity monitoring plan. Toxic safety monitoring or hence it is also referred to as safety monitoring or toxicity monitoring. Now, what is the difference between passive and active surveillance? So what the purpose of the passive surveillance is to assess the trends, to identify risk factors for its prevention and control, to get more and more knowledge about the adverse drug reactions, and also to identify the risk factors and how they can be prevented or how they can be planned. Then active surveillance is done to validate passive surveillance systems and to loosely track new invasions. So the active surveillance systems, or the studies which are planned under active surveillance, so they are also planned to validate the passive surveillance systems and to loosely track the new invasions. Okay, to track the new invasions, on to track the new adverse drug reactions which are reported. Then pros of the passive surveillance: it is inexpensive and can cover the large areas, because it is inexpensive, because it does not require any kind of labor work, any kind of professionals are not required. There's a form which is available online, and it is also available offline on pharmacovigilance centers, so that can be filled up and that can be submitted. Okay, so this is done only now. And the areas which are covered in this passive surveillance is like large areas can be covered, because information can come from any areas. Okay, but in active surveillance but on the other hand, what are the pros of active surveillance? That they provides the more accurate data and the complete data, and they also produce the data early and in timely manner. Then because when the adverse drug reporting is done, adverse reactions are reported, so there are many cases of missing information. Some information is presented, some information is given, some information is not given. So that is the passive. So that is the main so incomplete data. But in active surveillance, you have the complete data. The data is more accurate and verified, and also the data can be generated in timely manner. Then what are the cons of passive surveillance? That is, it is underreporting and incomplete data, as I said, because the forms are filled up, they are submitted. Maybe there are some information which is missing, which is very significant. Okay, without that information, the case cannot be assessed. Okay, so that is the case of underreporting or reporting the incomplete data. Then active surveillance can be resource intensive. Methodology must be well developed. So now here for doing this active surveillance, we require resources like third-party resources or in-house resources we require. And the methodology which is applied here in active surveillance, it should be well developed. Okay, so from passive surveillance and from active surveillance, the data can be collected. The next is cohort event monitoring. So in this approach, the monitoring study is prepared before the drug therapy even starts. A group of people are exposed to a drug for a predetermined amount of time and is closely monitored throughout the treatment. So cohort event monitoring is that when the participants, they are recruited in the study and they are exposed to a drug, and this study can start even before the drug therapy has been started. So drug therapy has been started. So here the group of people, cohort means group. Okay. So group of people, they are divided into 2 groups or 3 groups, whatever the study design is being followed. And the drug is exposed, the patients, they are exposed to, the participants, they are exposed to the drug for a predetermined amount of time, and they are closely monitored throughout the treatment. Monitoring is done for adverse drug interactions or those connected to one or more medications taken along with the target drug. So now in this monitoring, so monitoring is done to see drug-drug interaction. What are the adverse drug interactions? Means if the participants, they are taking 2 drugs, one drug which they are already taking and the other drug which they are taking along with that drug, drug which is under study. So where we want to see that what are the adverse drug interactions, whether the drug is interacting adversely with each other, which is maybe the chances of any adverse events or any toxic effect may happen. So that is majorly detected or examined in cohort event monitoring. Then targeted clinical investigations. So these type of studies are carried out to identify and define the negative effects of a medicine among certain populations, such as those with certain genetic abnormalities, pregnant women, and senior citizens. Okay, so these are the studies which are carried out to identify and to define that what are the negative effects of a medicine, okay, in a certain population. Like in a certain population, like for example, pregnant women are recruited in the study, and then what is the effect of that drug on a pregnant woman, that is being studied. Okay, so that is the targeted clinical investigation. So generally the targeted population can be the population with the same genetic abnormality, or pregnant women, or the geriatric patients, pediatric population with hepatic or renal impairment. Okay, so that is known as, so that particular population is considered for targeted clinical investigations, and the information related to the adverse effects which can occur in that particular population is the information can be collected. Okay, understood? Is it clear? Okay, then what are the steps which are involved in event case reporting, case processing, or which is also known as individual case, like ICSR, individual case reporting? Okay, so the workflow of pharmacovigilance case processing involves 3 key steps, that is data management, assessment of the relationship between the drug and ADR, adverse drug reaction, and timely reporting of all concerned to all concerned parties and regulatory authorities. So first, as the adverse effect is detected, so the data related to that adverse effect is input and is stored, and from the database also it is checked that whether such cases are already reported or not. So data management is required. Then there is if such cases are not reported earlier, so the relationship between the drug and that adverse drug reaction is being set up, so that it can be, or it is tried to associate the drug with the adverse drug reaction, so that it can be confirmed that this adverse drug reaction is not due to any other condition, it is due to that particular drug only. And then after that, the information is timely reported to all the concerned parties and the regulatory authorities. Now, what happens in the data management? ADR reports are gathered, pertinent information from the reports is checked and organized, and particular international codes are assigned to the names of the drugs and the ADRs. So for that, for assigning the coding is done, like the ICD-10 coding, or I guess nowadays ICD-11 is used. So ADR reports that is gathered, the adverse drug reaction reports are gathered, which are related to the information from the report, which are related to the information which is submitted in the report, and that information is checked and organized. And then after that, that information is coded and saved in the database. such as the code name is assigned to the name of the drugs, to the adverse event the code name is assigned, and that with those codes only the data is stored in the database. Okay. The next is assessment of the relationship between drug and ADR. So uh, that is known as the casualty assessment. So creating a connection between the suspected ADR and the suspected drug is the goal of this stage. In this process, resources like patient information, findings from the body of prior research, and outcomes of laboratory testing are utilized. So now what happens? First, the probable adverse event is detected. Okay, adverse event is detected, that is notified to the regulatory authority. Then the investigation or analysis is done about that adverse drug reaction, and then after that the casualty assessment is done. Means the casualty assessment, or the casualty assessment means the data, or is like the data is collected, and it is the data is collected, and there is a relationship which is set. On the basis of relationship is set, like whether this adverse drug reaction is related to a particular drug or not, and then the feedback is provided. Okay, so this is how this is the second step, that to set up a relationship between the drug and the ADR, or to understand that whether this drug is, or whether the adverse drug reaction which is reported, it is actually related to the adverse drug reaction or not, or it has been it is some other reaction or event which is maybe due to some other condition. Then timely reporting to all concerned parties and regulatory authority. So here, utilizing pharmacovigilance system as this is its multiple purpose, to determine if a medicine was responsible for a significant number of ADR instances, experts employ a variety of statistical indicators. So now what happens to utilize the pharmacovigilance system, the systems, the databases which are used for the purpose? So to determine, and from these systems, it is determined that whether the significant number of ADRs related to that particular disease has been reported or not. Okay, and for that, a variety of statistical indicators are used. Professionals promptly notify all parties involved and regulatory authorities if it is determined that a medicine has a significant number of ADR cases that have been recorded. So if from the database or from the pharmacovigilance system, it is found that this particular drug has previously also the same ADR has been reported. So that ADR has been reported. So that notification is sent to all the parties like marketing authorization holder and all other parties which are involved in this process. And also it is reported to the regulatory authorities. Okay, understood? Is it clear? Okay, so what happens when the adverse event occurs? So when the adverse events occurs, the patient or doctor submits the report to FDA or to the regulatory authority or to drug manufacturer. Generally, the reports are majorly submitted to the drug manufacturer, and rarely it is submitted to the regulatory authorities directly. Then after that, the drug manufacturer and the regulatory authorities, they reports the paper reports are processed into electronic form. So if they have processed, or if they have got the date reports in a paper form, that the same data is entered into the database and electronic form is generated. Then adverse event report data is entered into the FDA database. So in every country, they have their database for this. So for that individual country, the adverse event reports are then entered into that database. The report is reviewed by the safety evaluator at the FDA to find any unexpected safety issues with the drug. Then after that, when that adverse drug is reported in that database, a signal is generated. And then after that, the signal detection and signal assessment is done. A safety evaluator will evaluate that whether this evaluate or find that whether this adverse effect is related to the, or whether it is means in other cases also this report has been generated, or the same adverse event has been reported with the other drugs or not. And then on the basis of the information which is collected, the investigation is done to set up the relationship between that adverse drug reaction and the drug, and whatever the action is to be taken, that is taken by the regulatory authority.